So, okay, these slides are about the general introduction about quantum biology, right? So the idea is that, of course, in quantum mechanics, we don't know where is the border from the unrealistic world of a possibility of quantum mechanics and the objective world, let's say, classical-like, right? And the same questions we have in quantum biology. Where is the border? Nobody doubts that quantum mechanics has a structural role. We cannot explain chemical bonds without classical quantum mechanics. But whether there is a functional role, right? Given by quantum mechanics in biology, is the question of quantum biology. And of course, we want to use quantum mechanics to understand better biology, but also to create devices, right, that can be useful. So the origin of quantum biology, in a sense, it dates back, actually, but certainly this is a milestone in quantum biology, which is this book by Schrödinger, What is Life? No? And in this book, Schrödinger notes that a small amount of atoms, a few thousands, present in every cell, controls the macroscopic behavior of all the organism. So all the organism, the color of your eyes is coded in few thousand atoms. And so a living organism, even if are macroscopic, they don't obey statistical mechanics law. They obey, according to Schrödinger, mechanical law. So not order from disorder, but order from order. And according to Schrödinger, the origin of these mechanicistic clockwork was in quantum mechanics. For instance, he hypothesizes, way before they discovered DNA, we said, we believe in this book, a gene or perhaps the whole chromosome fiber to be an aperiodic solid, which in our terms would be a molecule. So what he understood is that quantum mechanical bounds explains the solidity of the genetic code and allows these to resist noise and to be, let's say, transmitted through generations, okay? And of course, when they won the Nobel Prize, they wrote to Schrödinger, the winner of the Nobel Prize, to recognize his contribution. But the point is that the strength of the coupling alone is not enough to explain a functional role of quantum biology, because when you have many elements, the density of state is very high, and temperature mixes all these states, right? So the question is, starting from here, is that can we have a functional role of quantum mechanics in large aggregates? Now, I will focus on two kinds of aggregates. I will focus on photosynthetic antenna, but also on macrotubules in the second part of the talk, okay? So Massimo will explain you much better. We will consider photosynthetic complexes on anaerobic bacteria because they are the most efficient in nature. And by efficient, I mean, I will tell you what I mean later, okay? But now this is the green sulfur bacteria, and these little things you see here are the chlorosome, right, Massimo? Are the chlorosome, which are the photosynthetic units of green sulfur bacteria. I will show you how they are done. And this is the purple bacteria. And this sphere here, Massimo taught me, these are the chromatophore, right? The photosynthetic unit of, I always misspell it, right? I call it the sphere, but he gets angry, you know. The little spheres, and he gets chromatophore, okay. And I mean, to make you understand how efficient these things are, take into account that green sulfur bacteria are found in deep sea with no light, and they can live out of the thermal energy of volcanoes under sea, 400 degrees. They are able to feed themselves with thermal energy. So these are an amazing systems. And so since light is very dilute, what they do, this photosynthetic antenna, both of plants and of bacteria, these photosynthetic systems are made of network of chlorophyll molecules that they need to collect light and to transfer the light to a reaction center, okay? So all these photosynthetic systems are made of networks of chlorophyll molecule. Here I'm representing a chlorophyll molecule with an arrow. You see, actually, a chlorophyll molecule is way more complicated than an arrow, right? It's made of hundreds of atoms. But there is this transition dipole moment Anna was talking about. The strongest one is in plane, is called QY, and this is the arrow I'm referring to, okay? This is basically the direction the electrons in the molecule oscillates when illuminated by the light, okay? There is also a QX transition, which is weaker, and so I'm focusing on the strongest one. So the excitation arrives, get transported to another complex, which is called the reaction center, and the reaction center gets charged. Here, the energy becomes an electron that moves, let's say, and it goes from the open state to a closed state when it's in a charged state, because the electron left the reaction center, and it stays in the closed state for about, let's say, 100 microseconds up to a millisecond and so on. Now, if you consider that due to the dipole strength of the chlorophyll, 10 photons per second are absorbed, and this remain closed 100 microseconds, it means that these antenna have to, from 100 microsecond to 1 millisecond, they need to feed this to enhance the ratio of the excitation reaching here by 1,000 times. And that's what the antenna do. They enhance the number of excitation that reach the RC by a factor of 2 or 3, okay? A factor, an order of magnitude, 2 or 3. And that's what they're built for. So when I say that purple bacteria and green are efficient, what does it mean? That every photon that is absorbed by the antenna goes into a charge-separated state in the reaction center. So the efficiency is almost, the internal efficiency is 100%. This doesn't mean that the overall efficiency, external overall efficiency, is 100%, because these antenna absorb few percent of the light, and moreover, you produce glucose with an overall efficiency of less than 1%. Okay? So photosynthesis is not an incredible machine, but this part, the transfer of the energy after absorption is an incredible, almost perfect machine, okay, nanoscopic machine. I mean, not the total efficiency, not the total efficiency.
You have way more than 10 photons per second as well, right, available in sunlight.
Yeah.
You've got 10 to the power of, I don't know, without checking, right? It's huge.
Yeah, yeah.
And a lot of that is not needed.
Yeah.
Right?
Yeah, I mean, yes, I mean, you absorb a few percent, 3% produced in chromatophore, and then 100% goes here, but then you lose in the chemical reaction. I don't know why. I mean, but it's enough to leave, so they stop there probably, right? But they wanted to make, I mean, these make sure that everything you absorb goes to the right place, right? So this is an open question, by the way, yeah? Why life, this part is so efficient and the rest.
I may surprise you with some information.
Yeah, maybe you can tell us something. Yeah. So now, in these photosynthetic units of purple and green sulfur bacteria, they have a very hierarchical and symmetric organization of the chlorophyll molecule in the antenna. They're not randomly put. For instance, we're starting with the chlorophyll molecules. In green sulfur bacteria, they are self-aggregated with specific direction of the dipole and very symmetric disposition in huge cylindrical nanotube, which are 200 nanometers long. Okay? And in the chlorosome, which is the stuff I showed you before, there are several concentric cylinders inside. They absorb the light. They transmit the light to another aggregation of chlorophyll molecules, a different kind of, called the base plate. Then below the base plate, there is the FMO, which are again a mixture of protein and chlorophyll molecules. And from there, they go to the reaction center, which is also a mixture of protein and chlorophyll molecules. Okay? That's the organization. And you have overall 10 to the 5, more than 10 to the 5 chlorophyll molecules in this system, okay? In purple bacteria, the chlorophyll are arranged in rings, like Anna has shown. So these purple stuff here are the chlorophyll molecules. Smaller rings are LH2, and then bigger rings is LH1, and then there is the reaction center inside. And you see, and these rings are arranged on a sphere, which is called the chromosome, which is what Anna showed, okay? Now, an open question is, what is the relation between this structure and functionality? And I will show you also here the direction of the dipole in these rings is not random at all. They have a very specific and symmetric structure, okay? So what is the relation between structure and functionality? And can quantum mechanics have a role in such large aggregates? We're talking 10 to the 5, 10 to the 3 chlorophyll molecules. Now, this is the slide that Anna showed before, so I will briefly repeat myself. But the point is that what I want to focus now in what is the relation between symmetry and cooperativity. Okay, so one point I will make is that all this symmetry here creates cooperative behavior in this antenna. Which cooperative behavior is the same cooperative behavior that Anna just mentioned. If I have two molecules, right, then with only one excitation, then if they interact through Foster, for instance, then this is the ground state, and you have two excited states. One is the excited state of one molecule minus the coupling J, and one is plus J. And then now you have one transition with a transient dipole moment mu, right? But now you can have two transitions from the ground state to this state or to this state. If this dipole is larger than this, then we say that this is an H aggregate, because the upper state is larger than the lower dipole moment. Vice versa, if the lower dipole is stronger, you have a J dimer, right? In general, symmetry creates cooperativity. What does it mean? Symmetry implies then when all these molecules interact, you have n energy levels, but the dipole strength from the ground state to each of these energy level is not uniformly distributed, but is concentrated in a specific energy, okay? If you have a random disposition of dipoles, then all these energy levels would have more or less the same transition dipole, okay? So that's what we call cooperativity in this case. In the extreme case, you can have one state here, which absorbs n times, who has a dipole which is n times larger than the single molecule. And this is called a giant dipole or a super-radiant state. And it means all the other states have a zero dipole, so these are sub-radiant or dark, okay? Just for terminology. Now, what are these? All this symmetry is needed to create this cooperativity. And so the question is, there are two questions, right? If we have this cooperativity, why it is useful? And two, whether these natural systems have cooperativity or not, okay? So as I said, when you have symmetry, you can have this cooperativity, so a state with a giant dipole. And what you can do with a state with a giant dipole, or in general, how cooperativity can donate robustness. So first of all, if you have cooperativity, you have a larger energy scale. So you have fast time scale. And so you can beat the phasing, right? Because you can create something which is faster than the phasing. And so you can have, superabsorption, I'm sorry, Eric, is an old slide. Now we agreed we don't use it for single excitation, but just for main excitation. But let's say you can have a fast absorption n times faster at that specific energy, right? But mainly, you can have super transfer, because if you have an aggregate who has a giant dipole, and it can transfer much faster to another aggregate who have another giant dipole, or it can transfer much faster inside the same aggregates if each part has a giant dipole, okay? So this is called super transfer, okay? Which can be really fast, and so you can beat the phasing in this way. And another effect of cooperativity that we discussed in a paper in which Alessia is involved, and actually we looked precisely at the nanotube of green sulfur bacteria, is that this super transfer, these green sulfur bacteria can be modeled as first approximation as made of stacked rings, okay? Each ring has a super-radiant state, and the super-radiant state in each ring coupled between them, okay, because of the symmetry of the system. Actually, each eigenstate of the ring couples only with the corresponding eigenstate of the other rings. And so you create a super transfer coupling between the super-radiant states in all the ring. And if you have a larger coupling, you have a smaller density of states. You understand? Because if your energy is larger, you have the same number of states, then the density goes down. If the density goes down, it means that the thermal effects are less strong. And that's why in this system, we have shown that you can have the excitation at room temperature can be coherently spread over the nanotube over hundreds of molecules. So to make you understand the point, if I take a random disposition of dipoles here, at room temperature, you would have that excitation is spread over 2 free molecules, okay? While with a symmetric disposition and with these specific supertransfer coupling, you have a suppression of the density of states, which is, let's say, shown in this graph, in which you see that the energy gap between the lowest excited state and the next in energy excited state, the energy gap increases with the size instead of decreasing with the size. So if you have emergence of energy gaps and a suppression of density, that creates a huge delocalization of the excitation in these structures, even at room temperature. Now, before showing what happens in these natural antenna, I have only one slide about what kind of Hamiltonian we use. And you see, Anna, we don't use the Frenkel Hamiltonian. What you have shown, this dipole-dipole interaction, is an approximation that works when the two molecules are very close, so in the small volume limit. Actually, we use let's say, the full-fledged antenna interaction. This is the same Hamiltonian you find in Jackson, in a sense. Because here you have charge that moves. They're like antenna. They interact, right? So this is called an effective non-Hermitian Hamiltonian, and is able to describe the system even when it's much larger than the wavelength, okay? It has no limitation in this sense. Yes?
I've read very carefully the paper from Celardo and Luca. So the problem is not the interaction between the molecule, which is dipolar approximation, it's the interaction with the light that you cannot use the dipolar approximation.
Yeah. I mean, it's because dipolar approximation can mean different things. Yes.
But not when you discuss the interaction with light.
Yes. Well, the point is that when a molecule is excited, you then re-emit the photon either virtually or really, and then the interaction that mediates with all the other molecules should be taken into account fully, let's say. I mean, in the small volume limit, so when the wavelength is much larger than the size, this reduces to the dipole coupling, okay? But this allows us to study, sorry for the coughing, the cooperative response of the whole antenna system, and now is what I will show you, okay? So let's start with a smaller system.
Just a question, can you go back to your assumption? The wavelength is much larger than the size.
No. What I say that this Hamiltonian works everywhere, whether it's much larger or much smaller. While the dipole approximation works only if the wavelength is much larger than the distance. Yes.
You're just mentioning that as a comment.
That's this Hamiltonian reduces to this when the wavelength is much larger than the size. Okay? Our Hamiltonian reduces to the dipole only when the wavelength is much larger than the system. When not, we use this, which is correct always. That's my point. Okay? Indeed, we use this. We don't use this. And we will use this as a comparison. Okay?
So I guess this is not the coupling of the light, right? This is the coupling
It's the coupling between the molecules mediated by the light, let's say. I mean, is a,
mediated by the light, but
yeah.
the
No.
dissipative kind of emission.
So that the wider comparison would be hard.
No, sorry, I missed this point, Eric.
This is the Hamiltonian term, right? You're not modeling the electromagnetic environment.
So let's say if you take, it depends on what maybe, but if you take, a system, you remember, we take a system two-level system coupled with a photon buff at zero temperature, and you do the Markov approximation and the weak coupling, then you get exactly this effective Hamiltonian in this sense. So I don't know if this you want to call it the coupling with the light, but it can, maybe we are thinking of something else. But in this sense, it can be derived microscopically from, you have a photon buff and you have independent dipoles and you compute this Hamiltonian, let's say. So let's have a look at these rings. This is, I think, LH1. This is this bigger ring. You see these arrows, and so now you see the specific structure that the dipole of the chlorophyll molecules have in this ring, right? They're not random at all. And indeed, they create, this is the ground state, this is the lowest excitonic state, which is dark, and then you have two degenerate superradiant states in the first excited state of the system, okay? Now, so this is a J aggregate in the sense that the superradiant state is close to the ground, to the lowest excited state. Now, why nature did this, right? And by the way, superradiance had been measured experimentally by Van Grondel in 1997 in these structures to be of the order of 3 at room temperature. So they have a dipole which is 3 times larger than a single molecule. Here, of course, these simulations are without environment. If you put environment, you decrease, but still the superradiance can be seen experimentally in the system. And to come to the question of Dimitri, right? Why you have a superradiant state that emits very fast, you absorb there, but then you also emit, so you should lose. But the point is that this giant dipole connects very well with the reaction center, okay? That's how these structures are built for. And so the transfer time from this state to the reaction center is much faster than the emission time. And so you are very effective at transferring.
Reaction center trap.
Exactly.
You have a hole there and everything comes here.
Yeah, but the point is that you have a super transfer between the superradiant state in the ring and the reaction center, which also has a which is made of a dimer, which is also superradiant, and is a J aggregate. Also, the reaction center is a special pair, which is a J aggregate. So these two giant dipole couples, so that the energy gets here and gets there before it is re-emitted. Okay. And also in green sulfur bacteria, experimentally, you find this superradiant state, and you see these are the simulation. Look at this graph of what we show. We take the green sulfur bacteria, we diagonalize it according to the Hamiltonian, and from any energy, in which here we wrote in units of wavelength, I am plotting the dipole strength of each transition. So you can see this is the monomer excitation, there is a red-shifted giant dipole, which is 4,000 times larger than the single monomer, okay, at 800, and this exactly corresponds to the experimental data of absorption, of a red-shifted absorption at 800 in the green sulfur bacteria, okay? So these structures have superradiant states, have large dipole, okay, in nature. Now, what we need to understand is why, and I told you the first thing is that super transfer, okay? It's very useful. Now recently, we published a paper with Alessia in which we were able to use this non-emitting Hamiltonian to simulate basically the whole structure, the whole antenna, okay? Not just a part, right? Because here I show that this single cylinder has superradiance. Here I show that this little ring has superradiance, right? But now we consider the whole structure of the chromatophore. So we took into account more than 10 to the 5 molecules. So here this is a single cylinder. This is complex A, the smaller one. Then we take 4 concentric cylinder, and then we take 3 adjacent 4 concentric cylinder. And these have 132,000 chlorophyll molecules. These have tens of thousands, and this is, I don't remember, 20,000, this is 10,000, something like this. And also for the purple bacteria, we consider the whole chromatophore, okay? We model the whole chromatophore. So we study, the superradiance of a small unit, complex A, which has these 10 LH2 plus an LH1 and 2 reaction center, and we go to the whole chromatophore. And our question was, now we can, right, study this, because we studied this with a full non-Hermitian Hamiltonian, and we compare it with the dipole Hamiltonian, etc. And the question is, are these structure, will we have a more cooperative response the larger the structure is? Because this is not trivial. You can have superradiance or cooperativity in a small portion, then you increase, the cooperativity doesn't increase. For instance, if I would surround this stuff by randomly disposed dipoles, you wouldn't see any increase of cooperativity with the size, okay? So the question is...
Does it scale with dimension or not?
Yes. Does it scale with the size and with the dimension? Yes. And so what we found, to make the story brief, is that as you increase the portion of the complex you look, the cooperativity increases. And also, you see, Anna, the non-Hermitian Hamiltonian gives different results from the dipole and the Hermitian Hamiltonian, at least in Grissulfobacteria in the largest size. For the nanotube, you can use the dipole, okay? And while differences start to emerge, the dipoles start to fail when you take 4 concentric cylinder, and then free adjacent, you see the non-Hermitian gives very different dipole distribution, okay? For the chromatophore, the free Hamiltonians give the same result. So in the case of the chromatophore, you can use the dipole, because it's small, right? It's only 60 nanometer. But still, in all these, and this is the conclusion of this part, you see, this is how the maximal superradiance or dipole strength increases with the size. In both cases, you have an enhanced superradiance with the size, and this would nobody have measured this. That's why it would be nice to see this. You see what I mean? This will be part of APACE publication. I mean, in the future, if they do the experiments, to test, because we have a deliberate about characterization of the antenna. This is.
Julian is looking at me. Who was Dave? Looking in the general direction. No, you weren't. Thank you. I took it personally.
Okay, so the other part of the talk is about another topic, which is about biology related to the brain and consciousness, okay, in a sense, in a very wide sense. But the story is very interesting, and we did some nice work similar to this also in this, and since it was aimed to everybody, so I put it here, but it's very brief. So, okay, there is a problem nobody knows what consciousness is and how the brain works, okay? An interesting thing, this was an idea of Michael Fisher, which is a very good condensed matter theorist, who's bipolar, and he takes lithium, okay? And then he wanted to understand, that's what he says in his talks. I met him, by the way. And he said, I wanted to understand how lithium works, no? And then he found an experiment with mouses in which they give 2 different kinds of isotope of lithium, and they behave very differently, the mice. So when you do experiments with mice, you usually look at experiments when they have babies, because you can understand how they change their behavior, right? So with one isotope of lithium, they were very careful with the babies. With another isotope of lithium, they were careless with the babies, okay? And then you say, this is not a chemical effect, he said, right? Because it's lithium, it's the same chemical stuff. What's the difference? And he said, well, there is a difference between lithium-7 and lithium-6, because the spin decoherence of lithium-7 is 10 seconds, at C7, yes, and that of lithium-6 is 5 minutes, okay? So, and if there is, so one of the open problems in consciousness is called the binding problem of consciousness, which is if we take into account the velocity of the electric signals in the brain, you cannot account for the fast conscious decision, which has to connect the perception of different parts of the brains in a very small amount of time, okay? This doesn't match with the electrical signaling velocity, which is very slow. So one idea is that quantum entanglement is there, and so on, okay? And so Michael Fisher's point is that what's the only source of entanglement in the brain is spin coherence, because it's the only thing that can survive at room temperature, okay? So there is a problem, of course, that now this spin coherence has to be shared among the brain, right? And how can be shared among the brain? And now, so in the axons of the neurons, there are these structures called the microtubules. Actually, microtubules are everywhere in the cells. They control the cell separation. They are the backbones, the skeleton of the cells. And also chemicals and actually molecular machine walk on them, literally walk on them. So they transport chemicals, okay? But there is an open problem. Our point, it was the following. These microtubules are made of many amino acids, are made of a basic unit called a tubulin dimer, alpha-beta tubulin. Inside this tubulin, there are many amino acids. In particular, there are 8 tryptophan amino acids. And the dipole strength of tryptophan is the same of chlorophyll, okay? it absorbs at 280 instead of 500, but it's the same, so in the UV. So I was wondering, is the arrangement of tryptophan in these microtubules ordered enough to create cooperativity? Okay, so we studied with the same non-Hermitian Hamiltonian the whole arrangement of a micron-long microtubule of tryptophan networks, and what we found is that there is a super-radiant state in the ground state in the UV in microtubules. Okay, and this was our first, let's say, discovery about this structure. Okay, so what is the super-radiant state doing in a microtubule? So the idea that is behind is that there are biophotons in the body, which are produced by oxidation or reaction that you know better than me what they are. Anyway, there are biophotons. Now, if these microtubules are able to intersect these biophotons, cooperativity can help to transport this excitation very far. So they could mediate the entanglement of spin entanglement in different neurons through these biophotons. That's a possibility, okay? But I didn't work on that. I just noted that there is a cooperative, a super-radiant state in microtubule, okay? Then we won a grant with the UK foundations, which is called the Guy Foundations, to make experiments to see superradiance in microtubules, okay? These experiments were done by Chergui in Switzerland, right? I think Lausanne technology, okay? And so what we study also there, we study mega networks of chlorophyll molecules. So we study tubulin, we study the microtubule, then we study the centriole, and we study bundles of microtubules which are similar to what you have in axons, okay, neuron axons. Actually, experimentally, we were able only to study tubulin and microtubule, but not these two. There were too much scattering, it was complicated, it's in the UV, okay, so they were not able. So we have simulations for these, but we don't have experimental data. We have experimental data for these, okay? So theoretically, what we find, for instance, in axons is that there is cooperativity. Also, when you add, like here we take 91 microtubules, okay, which mimic a bundle in human brain, in human axons. And we see that as the length increases, the superradiance in the ground, there is a superradiant state in the ground state, and its dipole strength increases, okay, with the length. So superradiance occurs at the level of a microtubule, but it's also the level of bundles of microtubules, which could mimic axons. And then here we do some scaling analysis and so on. But the most interesting thing are these experiments, and in order to see whether there is superradiance experimentally or not, we computed the quantum yield at room temperature. Why? Because if there is a superradiant state in the ground state, so say that you have a certain non-radiative decay, right? The quantum yield, what is the fluorescent quantum yield? Is how much light that you absorb is emitted, right? Now, if you have a single molecule, the quantum yield is 12% of tryptophan, okay? So that we take that as a reference. Now, if there is a superradiant state upon aggregation, and assuming the non-radiative decay does not increase or decrease upon aggregation, then if you have a superradiant state in the ground state, your quantum yield can increase, right? And this is something we can also do to test we could measure quantum yield to test the superradiance effect in the size. So theoretically, at room temperature, take into account that chlorophyll molecules interact the interaction of the order hundreds of centimeter minus one. Tryptophan interacts of the order of 10 centimeter minus one. So these are very weakly interacting, very fragile structure. Nevertheless, at room temperature, we saw theoretically, this is the theory, an increase of the quantum yield, very weak, you can say, from 0.12 of single tryptophan to 0.14, okay? Well, experimentally, they got the same value, 0.14, of quantum yield increase from tryptophan to microtubule, okay? So I don't want to say this is a proof of nothing, okay? They were very difficult experiments to do. I just want to say that there is at least a coincidence, okay? Let's say a coincidence between experiments and theory. Of course, it would be very nice to test larger structure, okay, to see this. But this is where we had to stop because the funding was over and we couldn't measure other things. But anyway, there is a video on YouTube on these experiments by Sabine Hofstadter. We got 1 million visualization. Unfortunately, we only have 40 citations. So this is physics today. Today is like this. You have 1 million of visualization and 10 citations, you know. This is okay, so to conclude, so it seems that cooperativity is a general feature in many biological structure. Its functional role is not very clear yet, but certainly can give super transfer. It can enhance the diffusion coefficient excitation. by depressing the density of states, it can enhance the coherent delocalization of the excitational structure, which can also help to transfer the energy. And of course, we can exploit cooperativity, and this is, right, what we want to do, one thing that we want to do in Apache project, okay?
Okay, thank you very much.